Hello friends, I am Dr. Sirajul Akfaisi. I welcome you in my YouTube channel. Today in this video, I am going to talk about disaccharide of carbohydrate. You know my friend, disaccharide or oligosaccharide contain 12 units or 10 units of monosaccharide. So if you have disaccharide means 2 units of monosaccharide. On hydrolysis, if any carbohydrate gives 2 units of monosaccharide, then that carbohydrate is known as disaccharide. Before moving one term, glycosidic linkage, is important to understand what is meaning of glycosidic linkage in carbohydrate chemistry. You know my friend, the linkage between two monosaccharide sugar is called glycosidic bond or linkage. Depending on the number of monosaccharide unit, oligosaccharide are classified as disaccharide means which upon hydrolysis yield 2 monosaccharides. For example, maltose, sucrose and lactose. Keep in mind, upon hydrolysis yield 2 monosaccharide, that is called disaccharide. और उसके एग्जाम्पल क्या है? माल्टोज एंड सुक्रोज. मीन्स सुक्रोज और माल्टोज में कितने यूनिट है मोनोसेक्राइड के? तो 2 यूनिट. मूविंग फॉरवर्ड, लाइक ट्राइसेक्राइड विच अपॉन हाइड्रोलिसिस yield 3 monosaccharides. For example, raffinose and rhamnose. Raffinose on hydrolysis give 3 monosaccharide. That's why raffinose belong to trisaccharide. So monosaccharide, disaccharide, trisaccharide, tetrasaccharide, pentasaccharide and so on means on hydrolysis it gives accordingly number of monosaccharide. Now friends, maltose, which is a disaccharide, is a type of carbohydrate that is also called maltobulose or malt sugar. The chemical or molecular formula of maltose is C12H22O11. It is a disaccharide formed from 2 units of alpha D glucose that are linked by an alpha 1,4 glycosidic bond. It is composed of carbon, hydrogen and oxygen atom. It is a white crystalline, odorless, sweet tasting powder. Maltose is a reducing sugar and undergoes mutarotation. Mutarotation is also very important term in carbohydrate, so we should understand what is the meaning of mutarotation and what phenomenon it is showing. And maltose is reducing sugar. This portion is also very important because in objective type questions it is present, it is asked at national level examination like GATE, JM and GATE examinations. So you can see the maltose is equal to glucose plus glucose. Means if you hydrolyze this maltose, you will get 2 unit of glucose unit. Now what is the source of maltose? It occurs naturally in germinating seeds and can also be prepared in the presence of the enzyme diastase by hydrolysis of starch. You know, in a starch, that is the polysaccharide, it contains the glucose. Now, structure of maltose. Chemically, as a chemist, or if you are interested in chemistry, we are always interested in structure of the molecules. So, what is the structure of maltose? The chemical or molecular formula of maltose is C12 हाइड्रोजन 22 O11, कार्बन इसमें 12 है, हाइड्रोजन 22 है और ऑक्सीजन 11. It is a disaccharide formed from 2 unit of अल्फा डी ग्लूकोज. सिर्फ हम ग्लूकोज जो बोलते हैं कि 2 मोलिक्यूल ग्लूकोज है, नहीं कैसा ग्लूकोज? सो डी ग्लूकोज. डी ग्लूकोज में भी अल्फ़ा और बीटा है, तो कैसा? तो अल्फ़ा डी ग्लूकोज. And this is important for the objective questions. A maltose contains alpha D glucose, beta D glucose, alpha and beta both, none of the above, like that. So alpha D glucose that are linked by an alpha 1,4 glycosidic bond. Isomaltose is an isomer of maltose, where the 2 unit of alpha D glucose are linked by an alpha 1,6 glycosidic bond. Now you can see the 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. So this is the 5 carbon or 6 member ring because oxygen is present in this ring, that is look like pyranose. So add carbon 1, hydrogen and another linkage, that is the 1 is linked with the another unit of alpha D glucose at 4 position. That's why we called it 1,4 glycosidic linkage, because both is the alpha, that is the alpha 1,4 glycosidic linkage. Now 2 position OH group is here, that one is deoxy sugar, hydroxy sugar. If you remove this oxygen, then deoxy sugar. If you remove add carbon 2 with the NH2, then that becomes the NH2. And amino sugar, which is important term for semester 2 students, can write short note on the following like amino sugar, glycosidic linkage and deoxy sugar. At 3 you can see the situation of OH and 4 and 5th CH2OH. This is the unit of alpha D glucose. This is another unit of alpha D glucose, and a numbering start here oxygen 1, here oxygen 1, and both unit attached with the 1/4 position. That's why it is called alpha 1,4 glycosidic linkage in structure of maltose. So my friend, maltose is a reducing sugar because one of its glucose unit has a free aldehyde group. That is very important. Why it is reducing? Because it giving the reactions of aldehyde, it indicating that one of the glucose unit has the free aldehyde group. On treating maltose with sulfuric acid, carbon dioxide, water and sulfur dioxide are formed as by the byproduct. You can see C12H22O11, that is the maltose, sucrose, and it reacts with the sulfuric acid of 24 molecules, and it gives 12 molecules of carbon dioxide, 35 molecules of water and 24 molecules of sulfur dioxide. Now hydrolysis of maltose results in the productions of ethanol and carbon dioxide. You can see here 4 molecules of ethanol and 4 molecules of carbon dioxide. The hydrolysis is reaction of maltose in the presence of the maltase enzyme give two molecules alpha D-glucose. You can see these two molecules is alpha D-glucose. You know the structure, molecular formula of glucose is C6H12O6. Now uses of maltose. What is the use of maltose? So as maltose is not as sweet as sucrose, it is not often used as a sweetener, but it is extensively used in the manufacturing of beer and industrial production of alcohol. During the barley malting process, maltose is used to add sweetness to the beer. It is used as a bulking agent and diluent in bakeries, soft drink, sweets, alcoholic beverages, and baby formula. My friend, the shelf life of food is increased by adding maltose. It provides energy for a variety of bodily functions, allowing us to carry out our day-to-day tasks. As maltose has a similar compaction properties to lactose, it can be used as substitute for lactose in the pharmaceutical industries. It is one of the nutrients that helps our body function effectively and allows us to carry out our day-to-day tasks. In human, maltose is broken down into two glucose molecule by various maltase enzyme, and they are broken down further to provide energy or stored as glycogen. You know, my friend, isomaltose is also maltose, but here glycosidic linkage is different, 1,6 linkage. So you can see this is the position of linkage. One glucose unit, another glucose unit, it is not attached with the 1,4, actually it is attached with the alpha 1,6 glycosidic linkage. So structure of maltose. So my friend, isomaltose is disaccharide comprised of the glucose which can be metabolized to produce chemical energy. Isomaltose can serve as a source of energy. It may be obtained from the digestion of a starch or food containing IMO or isomaltose. Most of the dietary isomaltose is not obtained naturally. So overall, you have learned in this video about the disaccharide. Disaccharide unit means after hydrolysis it gives 2 units of monosaccharide. Especially I have talked about the maltose. Maltose have the two types, 1,4 and 1,6. 1,4 linkage, that is the maltose, and 1,6 linkage, that is isomaltose. Thank you for watching.